Welcome back. This session features a series of short talks by students and postdocs at the FitzPatrick Institute of African Ornithology. The institute was established in 1960 by Cecily Niven in memory of her father, Sir Percy FitzPatrick, famous as the author of Jock of the Bushveld. The institute was incorporated into the University of Cape Town in 1973 and recognised as a National Centre of Excellence in 2004. Our research is divided into two main areas: understanding and conserving biodiversity. In addition to our ornithological studies, we also convene a course in conservation biology that's been training the next generation of conservation leaders from all over the world since 1992. The Institute continues to grow from its humble beginnings, and we recently held our 60th anniversary AGM. It was particularly gratifying to have Cecily's daughter-in-law, Marina Niven, attending the AGM. You can see her sitting at the front next to BirdLife South Africa CEO Mark Anderson. The series of talks reflect the broad range of research conducted at the FitzPatrick Institute. I'm not going to go through all of the talks. You can sit back and relax and enjoy them. All of them are short and concise and geared to a broad audience, so hopefully they'll be easy to engage with. If there's anything you'd like to follow up with or learn more about the Institute, please visit our website, www.fitzpatrick.uct.ac.za, where you can download our annual reports, copies of popular articles, and find links to the many students and their research.
Hello everyone, my name is Michaela Stoffberg and I'm a PhD student in the FitzPatrick Institute of African Ornithology. I'm studying hot birds in the city, and this presentation seeks to answer the question of whether junk food can buffer an urban bird from the effects of elevated temperature. This project is a collaboration between the Hot Birds Research Project and the Red-winged Starling Project. So as we all may know, global temperatures have been steadily rising over recent decades, and this change has been recognized as a potential major threat to global biodiversity. Extreme hot weather events have already caused mass mortalities in endotherms, and these temperature-related mortalities may be a more regular occurrence under future climate change projections, thus reiterating the need to understand how species will respond to elevated temperatures. However, an overwhelming majority of the literature on climate research tends to focus its efforts on relatively intact ecosystems, limiting our ability to understand how human-dominated or urban systems will respond in the face of climate warming. And these are important systems to consider because not only is the world becoming increasingly more urbanized, but because urbanization intensifies climate warming. This is due to a phenomenon known as the urban heat island effect. So the urban heat island effect results in the air and surface temperatures in highly urbanized environments being substantially higher than those observed in park and rural areas. And this is predominantly due to a high percentage cover of artificial surfaces such as bricks and concrete, which have a lower reflective and evaporative capacity compared to highly vegetated areas, resulting in a high amount of heat being stored and absorbed in the urban structure. Birds may be particularly vulnerable to the effects of elevated temperature due to their relatively high body temperatures, their small body sizes, and their predominantly diurnal habits. And so, to dissipate excess heat, birds can use one of two ways. They can respond physiologically by dissipating excess heat through panting or evaporative cooling. However, the capacity to use evaporative cooling is limited because evaporative cooling requires water and comes into direct conflict with the need to conserve water and avoid dehydration. And so to mitigate that cost, birds can use behavioral means of thermoregulation, such as using thermally buffered microsites or reducing overall activity. However, behavioral thermoregulation is also not without consequence. Studies have shown that these southern pied babblers on hot afternoons traded off their foraging against panting and were not able to gain enough food to sufficiently offset overnight mass loss. Similarly, these southern fiscals experienced a 50% reduction in their foraging success due to them shifting their hunting perches from these open, sunny perches to these shaded, more vegetated perches in return for the thermal benefit. As well as suffering from elevated temperatures within urban environments, urban birds also have potential benefits, one of which is access to greater anthropogenic food. And so this raises the question of whether or not, when you have access to greater anthropogenic food abundance and water resources in urban environments, can the costs associated with thermoregulation be mitigated or avoided? At the University of Cape Town campus, we have a system where we can potentially explore this because we have a population of red-winged starlings that is highly habituated to human presence and is often seen consuming human food on campus. So we hypothesize that urban birds benefit from abundant urban food resources such that thermoregulatory costs are mitigated. So we predict on high human presence days, such as weekdays and during the term, when there's lots of students on campus and as a result lots of junk food, that the costs associated with thermoregulation will be less severe. In contrast, on low human presence days, when there's less students on campus, such as weekends and during the vacation, and when there's less junk food, that the costs associated with thermoregulation will be more severe. We explore this using focal data, where we conduct 20-minute focal observations, recording the main behaviors such as panting and foraging of red-winged starlings, using the CyberTracker software. Also, we use mass data, where we record mass change throughout the day by weighing starlings in the morning and in the late afternoon. So looking at our first result, which is panting, we have the probability of panting on the y-axis and temperature on the x-axis. We also have our responses seen on high human presence days in orange and the responses observed on low human presence days in blue. So as we can see, the probability of panting increases significantly with temperature. However, these patterns are not significantly different between high human and low human presence days. Our second result looks at foraging effort. On the y-axis, we have the proportion of vocals spent foraging and again temperature on the x-axis. So as we can see, foraging declines significantly with temperature, but similar to our first result, these patterns are not significantly different between low human and high human presence days. Our next result is consumption rate, which is expressed as beakfuls consumed per minute focal on the y-axis. Our results show that despite lower foraging effort with temperature as seen in the previous slide, consumption rate of red-winged starlings is not significantly affected by temperature. And these patterns are also not significantly different between high human and low human presence days. Our final result looks at percentage mass gain. On the y-axis, we have diurnal body mass change. Similar to all our previous results, the pattern of diurnal mass change with temperature is not significantly different between high human and low human presence days. So our results don't show a difference in the thermoregulatory costs with temperature between high human and low human presence days as we predicted. So whether junk food can buffer birds from the effects of elevated temperature is not clear from our results, because the responses seen on low human presence days is similar to those seen on high human presence days. What is clear from our results is that despite increased panting and lower foraging effort with increasing temperature, the overall resource abundance in the urban environment is above the threshold at which these thermoregulatory efforts incur costs to food consumption and mass gain. This suggests that urban starlings within the non-breeding season don't seem threatened by current temperature conditions. Thank you for listening.
Greetings, everyone. I'm one of a couple of talks here today from the Hot Birds Research Project. It's an international research group focused primarily on understanding the biology of birds dealing with high temperatures and the threats that those birds face. A particular focus of the Hot Birds team and myself is, very obviously, hot, arid zones. As I'm sure you're aware, deserts can be very hot and dry, and they support some of the lowest primary productivity of any terrestrial ecosystem. Generally, they're just really extreme places for organisms to live. So with the climate now rapidly changing, we need to know how birds in these areas deal with their environmental challenges and how they might react to change. I work specifically on these bizarre and amazing birds. Yellow-billed hornbills are interesting and pertinent to the study of the biological effects of climate change for a number of reasons, but there's just two I want to highlight here. First, they breed in cavities, they forage primarily on the ground, and they breed in summer. All of that makes them particularly vulnerable to high temperatures and the low resource availability associated with high temperatures and drought. Second, they breed quite readily in nest boxes, which makes it possible for us to answer a lot of neat questions regarding their reproductive ecology, because we have this amazing access to the female and the nestlings inside the nest. Now, these are birds that actually seal themselves inside the nest cavity during the breeding attempt, probably to stop predation. So this is footage from a nest camera on a natural cavity, where you can see a genet having a sniff around. And this probably would have resulted in a predation event if the hornbills didn't have their amazing nesting strategy. Now, this graph essentially sums up probably the most pressing issue and why the Hotbirds Research Project is doing the work it does. This graph has the average daily maximum temperature between October and March each year on the y-axis for 3 weather stations in the Kalahari dating all the way back to 1960. Basically, you can clearly see that it's getting a lot hotter out there, which means that the birds which attempt to breed during and between October and March are starting to face increasingly difficult conditions. This is true for many seasonally hot arid regions around the world, so how it affects the organisms there is really the major concern. Now, if you look at these two graphs from the breeding population of hornbills that I study over the period of about a decade, each of these points represents an entire breeding season. So you're seeing 8 years worth of data here. In the top panel is the number of breeding attempts at my field site each year since 2008 up to 2018. And in the bottom panel is the mean number of fledglings produced per attempt. And that's over the same time period. Clearly, you can see that there's been a collapse in the breeding performance of this population of hornbills, and that corresponds to that increasing temperature I showed you in the previous graph. So, we have this collapse in the breeding success of the hornbills over the last decade, correlating to increased temperatures. But now we have to investigate what is actually driving that pattern. Now, my field site is one that goes through major seasonal change, from the dry, cool winter when nothing seems to grow, the ground is bare, through to a hot, rainy season in spring and summer. That eventually transforms the landscape into an unrecognizably green place, and many organisms, including the hornbills, only breed in the rainy season when resources become more abundant. Now, I raise this seasonality because it creates a situation where the highest resource abundance in the system coincides with the highest temperatures. Basically, the hornbills are constrained to breed when it's rained because they need the resources that arrive with the rain. But this creates a huge problem for the males, which have to attempt to provision to both the female and the nestlings during the hottest time of year. So provisioning and foraging effort for the males has to be traded off against their own thermoregulation. Also, the females are sealed inside the cavity with the eggs and the nestlings. So they're also very vulnerable to high temperatures because they don't have an option to escape or to forage for themselves. Now what you see here is a graph with the probability of a nesting attempt managing to successfully fledge at least one chick as a function of the average maximum temperature during the breeding attempt. So this is for relatively wet years, which is the solid black line, and for relatively dry years, which is the red dashed line. Essentially, dry years cause near ubiquitous failure regardless of the temperature, showing a really strong dependency on rainfall and the resources that arrive with the rain. But you can see that in relatively wet years, temperatures are having a really strong effect, with high temperatures during the breeding attempt drastically reducing the probability of a successful breeding attempt. Notably, you'll also see that above about 35.8 degrees Celsius, there's not a single successful breeding attempt in wet or dry years. And you'll see why that's so important in just a second. So this is a figure modified from a fellow Hotbirds Research Project member, Shannon Conradi, in a paper her and her colleagues published just last year. It shows the average summer maximum temperature under current conditions, and then under predicted future conditions by 2080 as you move to the right. The cross-hatching is the distribution of the hornbills. So as I just pointed out, remember, no successful breeding attempts above 35.8 during the attempt. The areas in dark red and gray are regions which will see average summer temperatures over 35 degrees Celsius by 2080. So according to this figure, we may see almost complete breeding failure of the entire Southern African population of southern yellow-billed hornbills by the end of the century. And that is where my field site is. So you can imagine that for the actual population of hornbills I've been studying, things are looking very dire indeed. So to sum up, myself and the Hotbirds Research Project are looking into the effects of high temperatures and resource availability on reproductive performance because the climate is now rapidly changing. Both temperature and resource availability affect the physiology and behavior of arid zone birds, and we're seeing that effect cause a drastic reproductive failure in the hornbills that may see them become locally extinct across much of the hottest parts of their range within the next 2 decades. So this story is obviously specific to the hornbills, but likely indicates a more widespread problem for many arid zone species, which are similarly constrained to breed in the hottest parts of the year in response to rainfall. And all that remains is for me to thank you very much for your attention.
Hello, my name is Amanda Bourne. I'm a PhD candidate at the FitzPatrick Institute for African Ornithology, where I work across 2 research projects, the Hotbirds Research Project studying the impacts of climate change on desert birds, and the Pied Babbler Research Project studying communication, cognition, and cooperation in cooperatively breeding birds such as southern pied babblers and Western Australian magpies. This slide just shows the wonderful team I've had the privilege of working with over the last 4 years, as well as all of the institutions that have funded our research. My research question is about whether group living can buffer against the impacts of climate change in cooperatively breeding bird species. I'm bringing together a theoretical question around what the benefits of group living are and why cooperative breeding may have originally evolved, together with a conservation question around what it is that underlies vulnerability to climate change in particular species. The question around whether group living could buffer against impacts of climate change emerges from comparative global research that has shown that cooperatively breeding species are more likely to occur in harsh environments. The association between cooperative breeding and harsh environments has been relatively untested in the field. And so with this study, we've been looking at the ways in which group size in cooperatively breeding birds interacts with the impacts of weather variables such as temperature and drought, in order to understand whether larger group size can moderate the effects of harsh weather. The study species we've been working with is the southern pied babbler. They are cooperatively breeding passerine endemic to the Kalahari. We have long-term data available for a study population in South Africa, which was established in 2003. And the birds in the study population are habituated to observation within a couple of meters, which means we can collect detailed behavior data from them. They're also individually marked so we can track them throughout their lives. We've been collecting lots of different kinds of data in order to address our research question, including detailed weather data from the study sites, as well as a lot of data from breeding groups. So we've been collecting daily growth rates of nestlings, monitoring nest outcomes, and then also collecting behavior data and weights from both breeding and non-breeding birds in groups with nests. We furthermore developed a non-invasive physiology technique for measuring metabolic rates and dehydration status in the birds without having to handle them. We show that hot nests are less likely to hatch. So when a clutch is incubated at ambient air temperatures above about 35 degrees on average, nests or clutches were half as likely to hatch as clutches which were incubated below that temperature. We also found that hot nestlings don't grow as well. So when we measured nestlings in the morning and the afternoon on hot days, we found that they grew less between the morning and afternoon measurement on hot days. And when temperatures were really warm in the high 30s and the low 40s, we frequently found that nestlings could actually lose mass during the day between that morning and afternoon measurement. We further show that hot nestlings don't reach fledging age. So in southern pied babblers, nestlings fledge 13 to 21 days after hatching. And we found that when ambient daily maximum air temperatures exceeded around 38 degrees, there was no successful breeding in our study population over 15 years. That is to say, no nest successfully fledged a chick at ambient temperatures above 38 degrees. We found that high temperatures also limited the survival of young babblers over winter. over their first winter to recruit into the adult population at a year of age. So when young babblers experienced high average daily maximum temperatures between fledging and nutritional independence at around 90 days, they were far less likely to survive over their first winter and recruit into the adult population. This pattern was exacerbated by droughts, such that in the coolest drought on record, we observed around 90% of young pied babblers surviving from 90 days through the first winter to recruit into the adult population at a year of age. But in the hottest drought on record, we saw only 12% of young babblers survive that first winter and recruit into the adult population. We observed a very similar pattern in breeding adults as well. So in the coolest drought on record, around 80% of breeding adults survived over winter and were present in the study population the following year, whereas in the hottest drought on record, around 30%. Only around 30% of breeding adults survived over winter and were present in the study population the following year. We tested whether group size or variation in group size would influence the effect of temperature and drought on all of the analyses I've just presented, and we found that group size never moderated weather effects. So in this example, showing the overall effect of temperature on overwinter survival in breeding adults, we show that variation in group size made very little difference in the effect that temperature was having on overwinter survival. In conclusion, high temperatures limited survival and reproduction in southern pied babblers, and this was evident across all life stages from egg to breeding adult. The effects of temperature were exacerbated by drought and were never moderated by group size. Given that temperatures are increasing at the study site and rainfall is becoming more unpredictable, resulting in an increased frequency of hot droughts, we are likely to see population declines and possibly localized extinctions in this species. Actions that we could take to ensure the survival of this species and other similar Kalahari birds, where we are seeing similar patterns in terms of the impacts of high temperatures and drought, are to reduce emissions, to slow the development and impacts of climate change, and also to conserve the habitat on which these birds depend. Thank you very much.
Hello everyone, my name is Kyle Middleton. I'm going to tell you a bit about my PhD project on individual contributions to group behavior in cooperatively breeding southern ground hornbills. So just a quick background on myself. I spent my early years in Gauteng, and after completing my undergrad, I moved to the Low Veld and was introduced to the FitzPatrick's APNR ground hornbill project. I joined them and been working with the birds for about 4.5 years now. For those of you who don't know where the APNR is, it's the northeastern part of South Africa, part of Greater Kruger, and consists of 5 privately owned reserves. Before I continue, I just want to thank everyone who's been involved, the Fitz and National Geographic for their funding, Mary Oppenheimer and Daughters Foundation for their generous donations, my supervisors Claire, Rita and Fanny, my assistant Carrie for always helping me, the APNR and everyone else. There's so many people. I just want to give thanks one and all. So about the birds. Southern ground hornbills, bird of the year. Hopefully you all know them, hopefully you all love them. They're these big black terrestrial birds which are unfortunately listed as endangered in South Africa. They live to about 50 to 60 years in the wild, and they are entirely frugivorous, eating everything from insects up to things about the size of hares. They're cooperative breeders, which means that the entire group contributes to the raising of offspring, and these groups consist of about 2 to 12 individuals made up of a single female and several male helpers of different ages. They occupy these large territories of about 100 square kilometers, and their productivity is very low, with a maximum of 1 chick being produced each season. So cooperation in general is quite interesting. It is group living, obviously, and is relatively common, yet it is intriguing because individuals contribute to the group benefit, but this comes at an individual cost. And when you look at the breeding, they forego their own reproduction to provide energetically costly help to others. This essentially makes us question its stability and wonder how it may have evolved. The way to understand this is to look at the costs and benefits associated with the different individuals, since evolution actually operates at the level of the individual. In simple terms, the benefits to the individual need to outweigh the costs, and that's the way to understand this. In order to analyze this and look at these costs and benefits, we need to actually look at who is contributing and how much. So the aim of my study is to investigate the social structures as well as understand how the different individuals contribute towards territory defense and reproduction, which are two vital group functions for survival. So ground hornbills are relatively well known for their booming morning vocalizations, since they use it as a means to advertise and defend their territories. But while they are well known, they've never actually been analyzed in detail, and little is known about who's contributing, what information they're conveying, how they are structurally organized, and what the functions are. We have been looking into this so far, and it appears that only the adults and occasionally the subadults contribute to this behavior. As for the calls themselves, they seem to indicate sex and carry signatures which can potentially allow for individual recognition. This is what we've found so far, with the final step being whether they can actually recognize one another. On top of this, we've been looking at the other vocalizations which they produce around the nest, and we have found a few. The contact call, the alarm call, the excitement call, and the begging and feeding call. The other side of my project is looking at the reproduction and in particular the provisioning. As I said earlier, the birds, or the groups rather, are made up of these different individuals, made up of several adults, subadults, and juveniles. So we're looking at how they are contributing. So inside the nest, the female alone will incubate, so they'll be feeding her or the actual chicks inside the nest. So we're looking at how much each of these individuals are contributing. And what we found so far is that the adults, again, are the ones who are doing most of the work in terms of feeding, while the subadults are doing less, but still doing something, and the juveniles are doing next to nothing. So the adults will feed about 3.3 times a day, the subadults about 1.5 times a day, and then the juveniles hardly ever. The other side of it is also looking with these feeding rates. Obviously, climate is bound to have an effect on a number of different biological processes. So we're looking at how the feeding rates might be affected by temperature. As you can imagine, in a cool morning or a cool day, the birds will be feeding at a relatively constant rate. However, when things get a bit warmer, the birds become a little bit more heat stressed, and so you'd imagine that these feeding rates slow down a bit. And this is exactly what we found. We found in this graph, you don't need to focus too much on it, but the red line just shows that the feeding rates actually decrease with increasing temperatures. So when you think about it, okay, so the feeding rates decrease with increasing temperatures. Do they then live in groups because this buffers these increased temperatures? So it makes you wonder, do more individuals contribute towards this? And to analyze that, we're using our long-term data to see if this is the case. So in conclusion, my PhD will improve our social system understanding of these very unusual birds and provide us with insights into the individual costs and benefits associated with these behaviors. It also gives us the basis to understanding cooperation and how it evolved in the species, and finally contribute towards the conservation of the species that's aimed at the reintroduction process. Thank you.
Hello everyone, my name is Chima Nwogu. I'm a postdoctoral researcher at the FitzPatrick Institute of African Ornithology, and I'll be talking to you about how black sparrowhawk nestlings prepare their immune system for rainy days. I work under the project Understanding Urban Raptor Populations. Rainfall is crucial for the timing of breeding in many species of birds. While some species breed in the wet season, others do so in the dry season. And it is not entirely clear why one species breeds in the wet season and another does so in the dry season. But there's also differences in rainfall pattern between environments. In the southwest of South Africa, where Cape Town is located, the rainfall is generally high but takes place during the winter, whereas in the northeast, the rainfall is generally low and takes place during the summer. So species breeding in these different regions are likely to face different environmental conditions if they breed at the same time in the year. The black sparrowhawk is particularly interesting because it breeds both in the wet season and dry season. But this is in Cape Town, where the first breeding of the species was recorded in 1993. In the historic range of the species in the northeast of South Africa, breeding takes place during the dry season, that is during the winter months. So by shifting its range from the northeast to the southwest, the nestlings of these species are now raised in a dramatically different condition from what they are raised in the northeast. And this can entail different challenges for the species, including food and pathogen pressure. What is interesting about the breeding season in Cape Town is that it's long. Laying starts from March to August, while the nestling period is from the end of May to November. So the nestlings raised during the peak of the wet season from May to August are raised in very wet condition, while those raised later are raised in drier condition. So within Cape Town, we are able to get a scenario that is similar to the difference between the population breeding in the northeast and the population breeding in Cape Town in the southwest. And from this, we can try to see how the difference in environmental condition can affect the physiology of these species. The wet season is likely to expose animals to higher infection risk due to higher vector abundance, higher pathogen abundance, lots of parasites, and contaminated water. And so animals in the wet season are likely to be more infected compared to those in the dry season. So for the black sparrowhawk, it is possible that nestlings raised in the wet season will face higher infection risk compared to those raised in the dry season. But the question is whether nestlings actually ramp up their immune function to deal with the higher pathogen risk that they may encounter in the wet season. I'll try to answer this question shortly. We collected blood samples from black sparrowhawk nestlings over 4 breeding seasons throughout the breeding season, that is, during high rainfall periods and during low rainfall periods, and measured 2 indices of innate immune function. What we found is that for the first immune index, where we tested the capacity of the immune system to destroy foreign cells, we found that this increased with rainfall. So the higher the rainfall, the stronger the immune function of nestling. We also found a similar pattern for the second immune index, where we tested the capacity of the immune system to destroy bacteria. So this means that black sparrowhawk nestlings strengthen their immune function with more rain in their environment. So why are we interested in variation in immune function of the black sparrowhawk? We see from this study that nestlings raised in wet environmental conditions have to invest more in their immune function. Although this might help them to withstand higher pathogen challenge and so increase their survival and may have facilitated the colonization of the wet breeding environment in the southwest of South Africa, it also means that the resources invested in immune function are no longer available to be invested in growth. So if there's no relative advantage of breeding in wet conditions, nestlings raised in drier environmental conditions are likely to have a selective advantage over those raised in wet conditions. But more broadly, we see that climate change and changes in rainfall pattern is likely to have a negative effect on species. So if species have to shift from breeding in wet or dry condition to breeding in new environmental conditions due to changes in rainfall pattern, they might face higher pathogen risk, and so may have to invest more in immune function. And we do not know whether all species are able to adjust their immune function in this way. So when we make plans for conservation, it's also important to incorporate how changes in environmental conditions are likely to lead to higher infection risk and increased immune cost, not just how it's likely to affect food availability and availability of nesting site for species. Thank you. I would like to acknowledge everyone who has contributed to this project over the last 2 decades. This project has benefited from numerous volunteers and students who have worked on the project over the years. And for everyone who has been listening to me, thank you very much, and please enjoy the rest of the session.
Hi everyone, thanks for tuning into the session. I'm Jaslina, and today I'll be telling you about some of my PhD research and how it was affected by the lockdown. In traditional conservation, cities have historically been shunned and seen as a lost cause. Unfortunately, as cities become the main engine behind global change and biodiversity loss, we can no longer ignore them. The reality is that over 50% of the world's population now lives in urban areas, and over 3% of the Earth's land area is covered by cities. This doesn't sound like a lot, but when you include the extended footprint of the land needed to feed these cities, it's closer to 50% of the Earth's surface that has been transformed. It's increasingly clear that we can't expect to put all our hopes on isolated pockets of protected areas in this sea of transformed land, and we now have to engage with the blurry areas in between where wildlife lives and where people live. But the flip side of this is that cities can actually hold significant biodiversity in between all the concrete, and we can greatly reduce the negative impact of urbanization by restoring biodiversity and ecosystem services to cities. Preserving green spaces within our cities is not only beneficial to biodiversity, but can mitigate climate change and also promote human well-being. Birds have become one of the most popular study subjects for understanding the health of urban ecosystems. The presence of a certain species in a city can reveal a lot about ecological function. By studying birds in human-dominated landscapes, we can understand broader ecosystem impacts, as well as more fine-scale mechanisms of how biodiversity responds to such extreme habitat transformation. The fact that birds are so visible relative to other more cryptic taxa makes them easy to study, but it also captures public interest in them. For these reasons, there has been an explosion of studies on urban birds and what they can tell us about ecology and evolution in the city. Although we now have a wealth of information on how birds respond to urbanization, the challenge remains that very few studies have been conducted in rapidly developing areas like Africa, and even from what we do know, cities are notoriously variable. Patterns of urbanization in the global south are completely different to the much older cities of the north. We need a better understanding of how biodiversity responds to this, so that we can come up with solutions that are appropriate for different cities around the world. And so this is one of the aims behind my PhD. The bulk of my research involves analyzing SABAP2 data from 7 South African cities and comparing urban bird communities with that of the surrounding area in order to understand why some cities are better at retaining more natural subsets of species than others. Looking at Cape Town as an example, we might expect that species such as these would be the most sensitive to urbanization and the first to disappear, whereas species like these are able to hang on and persist around the city's green spaces. And meanwhile, ubiquitous urban exploiters like these are able to thrive. The subup-2 pentads can reveal some of these patterns at a larger scale, but a separate component of my thesis involves understanding these dynamics at a much finer scale. In order to do this, I plan to conduct point counts around Cape Town, located along an urban gradient, with the intention of seeing where and why those more sensitive urban avoiding species drop off the map. While I managed to complete last year's fieldwork, this year was a different story. When it became clear that I wouldn't be doing much fieldwork this year, knowing that people would be homebound with a lot of time on their hands, we decided to change course and crowdsource these point counts instead. It would also free me from being restricted to Cape Town and from being unable to access private gardens. It was also an opportunity to collect data during this time and to see whether there would be any effect of the lockdown on wildlife. And so as the lockdown was announced, we shared this call on social media and we asked people to help by conducting point counts in their gardens. We asked people to provide 1 count per week of the lockdown, and we provided a survey to obtain some basic information about their property. The response we ended up getting was completely overwhelming. While we only planned for 1 count per person for 3 weeks, the lockdown soon became indefinite. Many people were so enthusiastic that they were submitting daily counts, sometimes even 2 a day, and some continued to do so until July. For many, the daily surveys became a source of routine and relief from the otherwise chaotic conditions. What started out as a plan to salvage a field season quickly turned into an impromptu citizen science project. At the peak of the lockdown, I was receiving over 100 emails a day containing spreadsheets and additional observations. Very quickly, I ended up with a vast and valuable dataset on my hands, but analyzing it all is something I'm still grappling with. While the point count data still need to be analyzed and land cover data extracted for each point, 256 out of the 282 who submitted data also filled out the survey, providing insights into features of gardens that might drive bird diversity and abundance. Most people conducted their counts from large gardens, though participants were based in a variety of properties. There was also a lot of variation in the number and maximum height of the trees in their gardens. We also asked people to record whether they were located in a complex or an eco-estate, but the majority of participants were in neither and were rather in independent houses. Participants were also asked to record whether they had pets, whether they had other cats that visit their property, and also whether they have squirrels or other predators that might visit their property. Of those who did have cats, we asked them to record whether they had an indoor or an outdoor cat, and we also asked them to give examples of some of the predators and other animals that might occur in their property occasionally. We asked participants to record whether they'd ever seen a bird nest in their garden, as well as whether they took any measures to attract birds, such as a bird bath or a bird feeder and indigenous plants. For some participants, the lockdown was an opportunity to make additional improvements to their garden, and many reported making changes to the amount or type of food they were providing. While the fact that we targeted birders means that these results are skewed towards wildlife-conscious residents, it shows that gardens can be a vital and underappreciated contribution to urban green space. While it's still too early to address my main research questions, what is clear is that the lockdown has served as urban ecology's time in the spotlight. People have appreciated the outdoors and urban nature more than ever during this time, and we can only hope that this renewed interest is carried forward so that our cities can be enjoyed by people and wildlife alike. Thanks for listening.
Hi everyone, this is Monique Duplessis speaking. I'm a master's student at the FitzPatrick Institute of African Ornithology, and my project is funded by SANBI. At the Fitz, I'm part of a research team that focuses on bird pollination in the Cape Fynbos. Specifically, we investigate how anthropogenic effects change the ecosystem services provided by nectarivorous birds. With my study, I assess how artificial nectar feeders affect bird-plant mutualisms in the Cape Fynbos. Wild bird feeding has already escalated to the point where it is essentially a global experiment. One type of bird feeding involves the use of artificial nectar feeders to attract nectarivorous birds to gardens. The costs and benefits of these feeders remain controversial, however. On the one hand, nectar feeders may have positive effects by attracting nectarivores towards suburbia, acting as stepping stones between fragmented natural areas, and supplementing their diets when floral resources are scarce. This may facilitate their adaptation to anthropogenic shifts such as climate and land use change. However, this may come at the cost of attracting them away from the plants they pollinate in neighboring natural vegetation, with consequences for seed set. It is therefore vital to understand the responses of nectarivore communities to these relatively novel urban sources, because it might affect all species involved in the pollination mutualism. To date, studies have only investigated the effects of nectar feeders on hummingbirds and bats in the Americas, with conflicting results. This is the first study to consider its effects on an African bird pollination system, despite the rapid expansion of urbanization in many countries. This study was conducted in the Cape Peninsula of the Cape Floristic region, where birds are important pollinators because a disproportionately large percentage of plant species depend on their pollination. I conducted experiments in Scarborough, Simonstown, Glencairn, and Clovelly, where nectar feeders are often available in gardens. I identified 18 sites across these 4 areas, each consisting of a residential garden on the urban edge, along with its bordering natural vegetation. I focused on 4 nectarivorous species that naturally occur in the Cape Peninsula, namely the orange-breasted sunbird, malachite sunbird, southern double-collared sunbird, and the Cape sugarbird. The endemic orange-breasted sunbird is of particular concern since it is almost exclusively associated with many bird-pollinated erica species. Erica species provide a useful system to test effects on pollination because their visitation rate is relatively easy to measure. For this study, I focused on the widely distributed Erica plucanetti and Erica abietina. The principal research question of this study was, do artificial nectar feeders in suburban gardens affect bird-plant interactions in the Cape fynbos? To answer this, I aimed to test whether nectar feeders affect local nectarivore abundance and distribution, and whether these changes in bird foraging and dispersal behavior affect local erica pollinator visitation rate. And so I hypothesized that nectar feeders attract nectarivores away from natural vegetation towards gardens, and that this disrupts the pollination processes of erica communities. To investigate the effects of nectar feeders on bird abundance and distribution, I conducted bird surveys at varying distances from gardens into the natural vegetation. At each site, I counted birds in 4 plots: the garden, 50 meters from the garden, 100 meters from the garden, and 150 meters from the garden. I alternated the presence and absence of feeders in these gardens, comparing bird abundance between sites with and without feeders. 20-minute bird counts were conducted in each plot, either in the morning or in the late afternoon. I also hypothesized that nectarivores may rely less on nectar feeders and more on natural flower nectar during winter, when floral resources are relatively abundant in natural vegetation. So I conducted bird surveys in winter and summer to test this. I also measured Erica plucanetti and Erica abietina pollinator visitation rate at varying distances from the edge of the gardens in the natural vegetation. At each site, I identified 1 to 6 patches of ericas, each patch containing between 1 to 6 erica plants. To measure the bird visitation of each plant, I inspected the anthering status. If the anthering was still closed, the flower was considered to be unvisited. If the anthering was broken, the flower was considered to be visited by a pollinator. And now for the results. When nectar feeders were absent, nectarivorous bird densities were greater in the natural vegetation and declined into gardens, suggesting that nectarivores generally avoid entering gardens. But when feeders were present, bird densities were much greater in gardens compared to gardens without feeders, and this was true for all 4 nectarivorous species. The attraction of the feeders was overall stronger than the aversion of the urban environment, even to sensitive bird species such as orange-breasted sunbirds. So nectar feeders may have positive effects for nectarivores by adjusting their urban sensitivity and facilitating their presence in suburbia. However, the presence of nectar feeders also decreased their densities in the natural vegetation. It is also important to note that nectar feeders attracted nectarivores over both seasons, regardless of floral abundance in natural vegetation. This finding is particularly important considering that feeders drew nectarivores away from natural vegetation. This suggests that nectarivores may be choosing to visit feeders over native flowers. With this in mind, it is possible that their adjustment in foraging and dispersal behavior is decreasing their visitation to local erica flowers. And indeed, what I found was that the presence of nectar feeders decreased Erica plucaneti visitation rate with approximately 11% across the 300-meter distance in the natural vegetation. In contrast, however, the visitation rate of Erica abiatina was not significantly influenced by feeders. So Erica species were affected differently by the presence of feeders. This may be due to differences in scent or nectar volume. However, the most likely explanation for the favoritism towards Erica abiatina may be that its handling time per flower is shorter compared to plucaneti, possibly owed to its shorter corolla tubes, which increases bird foraging efficiency. So in conclusion, the presence of nectar feeders attracted nectarivores away from the natural vegetation towards gardens during both winter and summer, thereby lowering their visitation to at least the local Erica plucaneti flowers, and potentially also lowering its pollen loads. The next step would then be to actually test whether this adjustment in visitation rate affects Erica plucaneti reproductive fitness. If this is the case, the nectar feeders could potentially increase local population declines along the urban edge in the long run. This study therefore demonstrates that supplementary bird feeding may have positive effects for birds themselves, but we also need to consider the negative cascading effects on local plant communities. While there are still a lot of questions about the effects of nectar feeders, we can contribute by making sure to clean our feeders regularly and by using only normal sugar, not sweetener or colorants.
Hello everyone, my name is Christian Brink. I'm a PhD student at the FitzPatrick Institute at the University of Cape Town. My larger project is about the role that vulture restaurants play in vulture conservation, but today I'm just going to be talking to you about the state of vulture restaurants in South Africa, which is important baseline information we need to answer our larger research questions. Now, these are some of the beautiful vulture species you can expect to find at a carcass in South Africa. Unfortunately, we're experiencing an African vulture crisis, and all of these species are currently at risk of extinction. Now, this is due to a range of threats, but by far the most acute threat is that of poisoning. Now, poisoning occurs both by livestock farmers putting out poisoned meat carcasses to target predators, as well as poachers that are specifically targeting vultures because they give away the location of their activities. And as you can see, one poisoning incident can kill a lot of vultures. So vulture conservation is quite hard because vultures range widely. They don't stick within the confines of protected areas. Vulture restaurants have therefore become quite a popular conservation tool to bolster vulture populations. Now, a vulture restaurant is basically just a specific site where NGOs or livestock farmers are providing meat for vultures to use. And the idea is that this safe food resource will also reduce the probability of vultures coming into contact with poisoned carcasses in the surrounding landscape. So first off, we wanted to know where are all the vulture restaurants, how much food is being provided at these sites, and we just wanted to know how aware vulture restaurant managers were of vulture threats, as this will feed indirectly to whether these sites are being managed in a responsible manner. To do this, we use data from various organizations involved in vulture conservation, such as EWT, KZN Wildlife, and Vulture. And we basically correlated all this information into a national vulture restaurant database. I then played telemarketer and phoned all the managers of all these sites and did a survey with them. So jumping straight into our results, this graph just shows the accumulation of active vulture restaurants through the years. And as you can see, this practice really started becoming popular in the 1970s and has grown since then, but in recent years has sort of plateaued. And by the end of a survey, we had a total of 143 verified active vulture restaurants across the country, with the main mentioned reason for starting these sites, of course, being to protect the vultures. So if we look at what's being fed, well, there's a huge amount of food being provided, more than 3,000 tons per year. Now this equates to about 83% of the food requirements of the entire vulture population in Swaziland, South Africa and Lesotho. Now we have to bear in mind that this food resource will also be shared with all other scavengers that have access to these sites. So if we look at the composition of this meat, we can see that the majority of what is being fed is beef, then followed by pork, and lastly game. So this map just shows the location of all the active vulture restaurants. The blue dots indicate them, and the size of the dot indicates the provisioning rate at these sites, and the yellow squares indicate sites that have already been closed. And then the gray shaded area indicates the species distribution of all those vultures I showed you at the start of the presentation. So there are just two important clusters of vulture restaurants I'd like to highlight, both of them encircled in red. Both of them are associated with important Cape vulture colonies, the first being up in the Magaliesberg and the second in the Molotsi-Drakensberg region. The second cluster in the Molotsi-Drakensberg region is also a very important conservation area for bearded vultures, and this area is sort of the last remaining stronghold of this species in southern Africa and is therefore very important. So we also asked managers about their perception of running a vulture restaurant, and 84% of them perceived some sort of benefit from running the vulture restaurant, with the most commonly named benefit being the cleaning service provided by vultures. Now, more than half also said that there's absolutely no disadvantage to running a vulture restaurant, with a few mentioning problems with increased problem animals such as jackal, the time and effort it took to maintain and clean the site, as well as some security issues regarding people coming onto their land to access the meat at vulture restaurants. To judge manager awareness, we asked them to list as many threats to vultures as they knew about. On average, they could only list 3 out of roughly 16 applicable threats. This means that awareness is quite low, and also awareness didn't exactly line up with how acute these threats were according to the literature. For instance, the most acute threat, poisoning, only ranked about 3rd, with less than 50% of managers mentioning this threat. There was also low awareness of threats that specifically pertain to vulture restaurants. And here I'm talking specifically about the threat of lead and veterinary drugs. Lead from spent ammunition in carcasses can cause lead poisoning in vultures, and the veterinary drug diclofenac has caused the complete collapse of Asian vulture populations. So they both have a food safety concern and can cause detrimental effects in vultures. So we asked managers specifically if they believed if either of these substances could have any negative health consequences for vultures, and only 32% of them agreed that lead can have negative effects for vultures, where awareness of veterinary drugs was much higher, with 72% of them agreeing that there could be some detrimental effects to vultures. So in conclusion, vulture restaurants potentially make a significant contribution to vulture diets in South Africa, but there's potential risks associated with these sites due to manager awareness being quite low, specifically as pertaining to food safety, with lead and veterinary drugs being a threat. It's therefore there is increased communication needed and training of people that potentially want to make use of this conservation tool. This brings me to the end of my talk. Thank you very much for listening. Thank you very much to all our collaborators. And if you want to know more, please follow the links to the relevant papers at the bottom of the page.
Hi, everybody. Thank you very much for tuning in. My name is Jeska van der Waal. I'm a postdoc researcher in the Honeyguide research project here at the FiTS. And today I will tell you a bit about our research and my projects on the ecology and evolution of the Honeyguide-human mutualism. This is a honey hunter from the Jawa tribe in northern Mozambique, and he's looking for bees' nests. But he's not very good at finding them alone, so he summoned the help of a little bird called the greater honey guide. This bird is also interested in the content of bees' nests, namely the wax. and it alerts the honey hunter of its presence by a specific chatter. And it then flies for typically a few hundred meters from tree to tree with the honey hunter following. The honey hunter will know where to stop because it's where the bird falls silent. And this is what we refer to as the Honeyguide-human mutualism. So when a honey hunter arrives at a tree that harbors a bee's nest, he will use smoke to subdue the bees and tools such as an axe to break open the nest, from which the honey hunters get their honey, and the honey guide gets its wax. So when we talk about a mutualism, we mean an interaction between two species where both partners benefit from it. So here the bird is providing information about the whereabouts of bee nests, and the humans are providing the skills to access the content of the bees' nests. So the greater honey guide is common throughout most of sub-Saharan Africa, and this remarkable mutualism between a wild bird and our own species is thought to be ancient, probably as old, if not older, than our own species. Once probably quite common throughout the continent, it is now much reduced and only occurs in certain parts of the continent. So one thing that fascinates us is to think about how the mutualism varies with human culture. Firstly, we can imagine that the skills a human has is important from the honey guide's point of view, as this determines how useful a human is to the birds. Secondly, the payoffs for the birds might differ between cultural groups. Some cultures actively reward the birds, while others believe the birds must be kept hungry. And thirdly, the human calls or signals that are used to attract the birds will differ between geographical areas. So there are only a handful of places left in Africa where the mutualism really thrives. And what I mean with thrive is that in these areas, humans rely on the bird to find wild honey, and selling this honey is an important source of income. Together with our collaborators, we study the mutualism in two areas, in Tanzania and in northern Mozambique, with the Hadza people and the Jawa people respectively. And there are some interesting behavioral and cultural differences between these ethnic groups that are relevant to the bird. One of these is the call that they use to attract the birds, which you will be able to listen to now. So what Claire Spottiswoode and Brian Wood have done in an experiment is record these different calls from these different areas and then swap them, showing that honey guides know their local human culture, meaning that honey guides respond more to the call that they are used to than to a foreign call. This shows that honey guides are adapting their behavior to these variations in human culture across geographical scales. What I was then interested in is exploring how cultural variation relevant to the bird varied within a cultural group, so on a smaller spatial scale. So for that, I interviewed honey hunters from the Jawa tribe in 13 different honey hunting communities across Niassa Reserve, which is twice the size of Kruger National Park. I asked honey hunters in these communities about their behavior relevant to the bird, as well as record the calls that they use to attract honey guides. I interviewed a total of 141 honey hunters, of which all but 2 were men, and everybody said that they use the greater honey guide to find bees' nests. The calls that they use to attract the birds are passed on from father to son. Despite the main calls being used differ across the villages, the mutualism still thrives in all the honey hunting communities I visited. Furthermore, we are interested to find out what is happening in the rest of sub-Saharan Africa, in these places where the humans have lost interest to interact with the honey guides. How has that affected honey guide behavior? Are the birds still trying to guide humans or not? So to be able to map the current extent of the guiding behavior, we recently launched honeyguiding.me, where I invite all bird enthusiasts across Africa to share their records of greater honey guides, specifying whether they were guided or not. Please visit our website for more information about honey guides and the project, and our website is available in English, French, and Portuguese. Our project poster describes in 4 simple steps how you can contribute as a participant in our citizen science project. We would like to know the location of the honey guide you saw, whether it was an adult or juvenile if you know this, whether you were guided or not, and we also welcome any additional information such as photos or behavioral observations, but this is optional. So these are only a few aspects of our research in the Honeyguide research project. We are 5 researchers based at the University of Cambridge as well as at the FitzPatrick Institute of African Ornithology. And if you'd like to read more about our research, please visit africanhoneyguides.com. And if you'd like to participate or read more about our citizen science project, please visit honeyguiding.me. Our work would not be possible without the amazing honey hunting community in Bamba village, as well as our partners and the European Research Council that funds our research. Thank you very much for your attention, and I hope you enjoy the rest of the virtual bird fair.
Hello everyone, my name is Ben Dilly. I'm going to be talking very briefly about our research on the wilkins bunting. So our study site is a tiny island. It's in the middle of the South Atlantic Ocean called Nightingale Island, which is the smallest of 3 islands which make up the Tristan da Cunha group. Now the buntings evolved from vagrant finches which reached the islands from South America, presumably blown there by the prevailing westerlies, and they've since radiated into 3 species and 2 ecologically distinct forms, similar to the famous and well-studied Galapagos finch radiation. The finches evolved independently at each of the 3 islands in the Tristan group, but the species at the main island of Tristan went extinct during the 19th century. And whereas on Inaccessible Island, the greater diversity of habitat supports 2 species which hybridize in some parts of the island, the habitat isn't as diverse, and the dominant habitat is tussock, which supports the small-billed generalist feeder, and the specialist large-billed wilkins finch is reliant on the woody fruits of the scattered phylica trees. So you might be thinking, well, what's so special about this wilkins bunting and this little bird? Well, the islands belong to the United Kingdom and are one of their overseas territories. So as part of their conservation of global biodiversity plan, the wilkins bunting was flagged as a species needing investigation, and as very little was known about it and about their population or what threats they faced, it's also known and is one of the world's naturally rarest species. It relies on only 10 hectares of forest. It's about 20 football fields on this one tiny island, and so it's listed by IUCN as an endangered species. You may also be wondering, well, how did we research these birds on such a remote island? And one of the core pieces of information you need to be able to begin to conserve any species is some idea of the population size, and so this is one of our main goals. So the only way to get there, to this uninhabited island, is by ship. If we're lucky, we go on the luxurious SA Agulhas, the South African research vessel, but sometimes we go on little fishing vessels which are not quite as luxurious. And this is how the island looks most of the time. The weather is very changeable. It's very tiny. It's about 2 by 2 kilometers, and this is where we stayed for 5 months at a time, completely off-grid, catching rainwater, surviving off a solar panel. And the buntings are reliant on the forests. So we searched for birds and began to catch them with hand nets and with mist nets. And then we color banded birds so we could identify pairs with binoculars and resights. It was slow going. Catching more than one new bird in a day was reason to celebrate. The island, this is a map of the island. The green indicates the copses of Phylica, and the pale cream color is the tussock. So by resighting color-ringed birds, we slowly built up a picture of each pair's territory. Over time, and the map filled out until once done, we estimated there were about 120 pairs and upwards of 300 mature birds. So this was good news. It was higher than expected. And we also followed their breeding performance, and most of the nest failures were from severe storms or when endemic thrushes ate their eggs or chicks. And despite this poor breeding success, the population appears to be fairly stable and is restricted by the available Phylica forest. The most severe threat to this tiny population is when we discovered an invasive scale insect and sooty mold which are killing the trees. Now the invasive insect ingests large amounts of honeydew which support this sooty mold. The mold covers the plant surfaces thickly, restricting photosynthesis, which combined with sap loss can cause tree dieback and tree mortality. On neighbouring Inaccessible Island, the sooty mold drastically reduces fruit loads and causes the deaths of large mature trees, which are most important for these large-billed wilkins finches to feed off. So fortunately there may be a solution. There's a parasitoid wasps which are used successfully in agricultural settings to control the scale insects. The application on a remote island is somewhat more difficult. It will mean considerable planning and study, but there have been some success stories on the Galapagos Islands. So biocontrol is something that's being looked at, but until then we're proposing to BirdLife that the wilkins bunting is uplisted to a critically endangered species. Thank you for listening, and cheerio.
Hello, my name is Anthony Lowney, and I study the importance of social weaver colonies to the surrounding animal communities. I'm trying to determine what species use these structures, when they use them, and why they use them, but more excitingly, how these different species interact when they use a colony at the same time. Today I'm going to discuss one of these interactions and explain how Kalahari tree skinks eavesdrop on social weavers to reduce the chance of being predated by pygmy falcons. Social weavers build huge colonies, and these can contain hundreds of nesting chambers and house nearly twice as many individual weavers. Many other animals, both avian and non-avian, also use these structures. One such species is a Kalahari tree skink. These skinks are found in greater numbers on trees with colonies and trees without. This is independent of the size of the tree. Another species that strongly associates with weaver colonies is the African pygmy falcon. These falcons do not build their own nest, and in southern Africa rely solely on weaver colonies for roosting and breeding. They simply evict weavers from a chamber and use this chamber for its own purposes. The main diet of these falcons is small lizards, including skinks. But the presence of a falcon at a colony does not reduce the number of skinks that you'd expect to find here, even though these skinks are more likely to encounter falcons more often and therefore increase their likelihood of predation. So how do these skinks manage this perceived increase in predation threat? Well, falcons also prey on weavers, so it's predictable that the weaver's behaviour will change, becoming more agitated when falcons are nearby. Therefore, skinks may be able to use the behaviour of weavers to determine when predators nearby or when the coast is clear and it is safe to venture away from the safety of its refuge. For this part of our study, we decided to count skinks at colony trees when weavers were present and behaving in a non-threatened manner, weaving, provisioning and chattering. This would suggest that falcons are not currently present. We would then compare this to counts at the same colonies when weavers were aware, and therefore skinks would be less likely to gauge whether a predator is nearby and as a result remain in a safer location and would be harder to see. So here we counted twice as many skinks at colony trees when weavers were present than when they were aware. We also found 3 times as many skinks foraging on the floor away from the safety of the tree when weavers were present. These results suggest that when weavers are in the vicinity of a colony and behaving in a relaxed manner, then skinks are more likely to bask in the open and forage in riskier habitats than when weavers are not around. The next part of this study was to determine if skinks cueing weavers responded to approaching predators and then flee to cover. Here a predator, me, approached skinks at trees when weavers were present and other skinks when weavers were absent. When a skink ran for cover, I dropped a marker that could be used to determine at what distance a predator was from the skink when the skink ran for cover. On the left we have the mean distance in meters between the skink and the predator when the skink ran for cover, and along the bottom whether weavers were present or not. We found that the predator could get much closer to the skink when the weavers were not present, likely increasing the chances of the skink being caught. Additionally, of the 34 trials that featured weavers, both skinks and weavers fled at the same time. During the other 11 trials, the weavers always went first. This suggests that weavers respond to an approaching predator and then skinks respond to the weavers. Our final test was to determine if skinks can eavesdrop on weaver alarm calls towards approaching falcons and then flee for cover. To do this, we played 30-second recordings of both weaver alarm calls and weaver normal chattel calls to skinks at colony and non-colony trees. This was carried out when no weavers were present. We then observed the changes in vigilance and determined the likelihood of skinks fleeing for cover when presented with either of these calls. Here are the different recordings we used. The first is the normal chatter. And this is the alarm calls. For the first part of these results, we looked at the changes in vigilance at colony trees to both recordings. On the left is the mean number of head movements used as a sign of vigilance. The more head movements, the more the skinks were looking around, the more vigilant they would be. We compared the number of head movements during the 30-second recordings against the 30 seconds prior to the recordings being played. And this is what we found. You can see that the skinks at colony trees became much more vigilant when an alarm call was being played. But there was very little change in behaviour when the chatter call was played. So next was to look at skinks at non-colony trees. Here again we found an increase in vigilance when skinks heard the alarm call. However, unlike at colony trees, vigilance also increased when the chatter call was played. Next we observed whether skinks run for cover when they hear any of these recordings. So on the y-axis we have the probability that a skink flees to a refuge. This again was carried out on skinks at colony and non-colony trees. So at colony trees you can see that the likelihood of a skink fleeing for cover when presented with an alarm call is very high, whereas presented with the chatter recording at the bottom, that little green bar is the probability of them going. You can see that's very low, that they very rarely run for cover. And here at non-colony trees, although you can hardly see it, skinks are very highly unlikely to flee for cover when presented with either of the two recordings. So to conclude, when in the presence of weavers, skinks are more active and are more likely to bask in the open and forage away from the tree. They also use weavers as a warning for approaching predators, and they can then retreat to the safety of a tree cavity or crevice. They use the many pairs of weaver eyes, sometimes hundreds, as lookouts, and use their vocalizations as a warning signal. The difference in behaviour between skinks at colony and non-colony trees suggests that this behaviour is learned, allowing skinks at colony trees to escape to cover on hearing an alarm and not waste time looking for predators when weavers are chattering nearby. And these are my acknowledgements.
My name is Carla DeToy. I'm a PhD student at the FitzPatrick Institute, and today I will be talking to you about some of my research on the sensory ecology of probe foraging birds. So probe foraging is a method of acquiring buried prey used by various different types of birds. Now these birds can detect their prey in two different ways, either via direct or remote touch. Now direct touch is used by birds such as oyster catchers, and that is when the exact location of the prey item is only known when it comes into direct contact with the surface of the beak. Whereas remote touch, which is used by other birds such as ibises, enables the birds to actually detect their prey and where it is while it's still at a distance from the beak. And this is the sensory modality that I am studying and will be talking to you about today. How does remote touch work? Well, you have a little invertebrate moving around in the soil, and this creates mechanical vibrations in the sediment. Then we have our probe foraging bird coming along and probing its beak into the soil. And so then when a prey item moves around and creates these vibrations, they come into contact with the surface of the bird's beak. And these birds have a specialized sensory organ at the tip of their beak, very creatively named the Vulture organ, which is able to detect these vibrations, and the birds are actually able to sense, hey, there's something down there that I can eat, and are able to then locate and capture it. So this slide is showing a general overview of modern birds, and there are three different families that are able to use remote touch to locate buried prey. The scarlet-passed shorebirds, such as sandpipers and godwits, the ibises, and the kiwi from New Zealand. And despite the fact that these three families are quite distantly related, they all share this Vulture organ that enables them to use remote touch to locate buried prey. And this Vulture organ is characterized by hundreds and thousands of little pits or holes in the bones of their beaks. And this is a beak of an ibis. And inside these pits are actually specialized little mechanoreceptors that are able to detect the mechanical vibrations in the sediment as the birds are foraging. But today I'll be talking to you guys mostly about ibises, which are the group that I am studying for my thesis. So some of the observations in ibises that led to the formation of my thesis. Firstly, there is a huge amount of between-species variation in the structure of the Vulture organ. So here we can see the beaks of three different species of ibises, and the little holes or pits on the beaks are arranged in various different patterns, and they have different numbers on their beaks depending on the species. And what's really interesting is that some of the differences in the Vulture organ seem to be linked to habitat usage. So what this graph is showing here on the y-axis is the number of sensory pits on the beak bones, in other words a measure of the size of the Vulture organ. On the x-axis we have habitat index, which is just a general measure of how aquatic the habitat is that the birds live in, with 5 being completely aquatic and 0 being completely arid. So this is data from various different species of ibises from all over the world, and what this pattern is showing is that in species that tend to live in wetter habitats seem to have larger Vulture organs. And it is this pattern that we are trying to understand and which I am studying, specifically looking at the 3 common species of ibis in southern Africa, from the mainly terrestrial hardy-dow ibis to the mainly aquatic glossy ibis, with the sacred ibis falling as a generalist somewhere in between. So one of the things that we found is that there seemed to be a link between the soil moisture content of the sediment in which the birds are probing and the morphology of the Vulture organ, and that the Vulture organ size correlates with the moisture content or how much water there is in the sediment in which they are probing. So what we found is that species which probe in wetter soils with higher soil moisture content tend to have a higher number of sensory pits on their beak bones. And we believe that this pattern has emerged due to the different transmission of the mechanical vibrations coming from their prey items under different soil moisture conditions, as well as the species' ability to differentiate between signals coming from their prey items and noise from external stimuli in the sediment. We also found various correlations between different types of behavior and changes in morphology of the Vulture organ. Firstly, we found the Vulture organ size tends to correlate with the success of probing. So on the y-axis here we have probing success, which means the percentage of the probes attempted by the birds while we were observing them that resulted in successful prey capture. And we found that the species with larger built-up organs tended to have a higher relative success of probing. And this could be a good indication that they have a higher need for their built-up organ and use of probing and remote touch to locate prey, as opposed to using other senses and other foraging techniques like scavenging or gleaning things using visual cues. So because the built-up organs are characterized by those little holes or pits in the beak bones, they have a higher potential of being fossilized than, say, soft tissues do. And so what we can do is look at the beaks of fossil birds displaying these characters and hypothesize various different things, such as whether they were likely to be probe foragers that used remote touch to locate their prey, the relative importance of probing as a foraging mechanism to these birds, different aspects of their probing behavior, and the sediments in which they probe, which is also interesting because it tells us a little something about the habitats in which they lived millions of years ago. One of the groups of fossil birds that I am studying are the lithornithids, which went extinct about 45 million years ago. If we have a look at the top right-hand corner, we can see the fossilized beak of a lithornithid, and despite the fact that this specimen is millions and millions of years old, we can still see the many little pits in the beak bones that are preserved. And based on our analysis of lithornithid beaks, as well as a sample of modern birds, we've been able to determine that these birds were in fact able to use remote touch. And this is really remarkable because it shows that this unique avian sensory modality evolved millions and millions of years ago, potentially even at the same time that the non-avian dinosaurs were still alive. Finally, I would just like to thank the following organizations and people for their support throughout my thesis, and a special thanks to BirdLife South Africa and the FitzPatrick for letting me be part of this bird fair, and to you watchers for being willing to listen to what us young researchers have to say about our work on birds. Thank you.